I won't go into the few hormones in them. Then your thyroid, parathyroid, islets of pancreas is for insulin. Adrenal secretes lots of hormones like mineralocorticoids and glucocorticoids. They are involved in the regulation of lots of things like kidney functions and things like that. Testes, that's going to have androgen. Ovaries will have the estrogen, and sometimes these placentas also secrete hormones. So what happens is, so I'm going fast because this work on ovaries and make ovaries secrete other hormones. They have a thyroid releasing hormone or stimulating hormone which affects thyroids. ACTH, adrenocorticotropic hormone, which does the adrenal cortex. And then if you have a removal of or hypothalamus, if you have a removal of hypothalamus, so you don't have the pituitary inhibitory factor, so the prolactin is going to be secreting like crazy. Now that is very important because that's how all the prolactin research was done by they used to work when I was in Berkeley. They used to work on fish called tilapia, and tilapia they were removing lots of the pituitaries from them and have the prolactin secretion go wild. Okay. These are the functions, major functions. Growth hormones function, hormone that affects on the, and then these are classified as hormones by some people. Some people don't consider them. I won't have time to tell stories today. But usually what happens is, when I was a student, the biggest thing was to do the steroid receptors in the cytoplasm, in the cytosol. So what you do is, we had like patient sample come or whatever, the samples are coming, you measure the receptor in the cytoplasm. And there are several ways of doing, and I cannot go into the details of that, but sucrose density gradients, or they do the scattered analysis, tell you the number of sites and all that. And then there was a friend of mine called Bill King. He moved as a postdoc to University of Chicago, and he showed when he was doing the immunohistochemistry that everything was in the nuclei. So the theory was that when you are homogenizing the tissue, you are really releasing all the receptors back into the cytoplasm. That's why everybody was looking at the cytoplasm. Now BP was my paper first. So they bind to the cell surface receptor, and then the intracellular receptors are working on the steroids. Okay, so that's in the nucleus. It says cytoplasm, but it is nuclear. This person must have been working on transfers the signal into the cell using a second messenger system, and then it alters its activity into the cell. Now in case of estrogen signaling occurs. It will go all the way to the nuclei, bind to the receptor in the nuclei, and you have estrogen response elements which regulate the function of the estrogen receptor that binds to EREs by tyrosine kinases and TGF beta serine 309 kinase. Remember I told you. And then it goes into the nuclei and starts working on that. Okay, so there are some of the structures. The original ring, the rings are numbered like this. So it goes like this: 5, 6, 7, 8, and so on. Then circles up, goes down, goes like this. And so the ring is called cyclopentano, perhydro. So chemists know better than me, but that's the ring that's used for the steroid hormones. All the steroid hormones have basically this 4-ring structure. The fourth one is the 5-carbon ring. And if you look at the derivations, then they all start from cholesterol, and then from cholesterol it gets to the pregnenolone through all the way, and you get the aldosterone. Aldosterone is a mineralocorticoid, regulates the kidney function. And then if you look at this one here, and I'll take out some of the ones which are important in the next slide. I'm just showing you a global picture of steroid metabolism here, that many hormones that we work on, like estrogen and androgen, or many of the glucocorticoids, they are interconnected. So this minor difference in the hydroxyl group can make a testosterone and estrogen, okay, something like that. But it's all metabolism, so that's how it works. These are some of the structures of the more known steroid hormones. This is how the progesterone looks like, and the cortisol, testosterone, and estradiol. The only thing that you want to remember is estradiol and testosterone, right? I mean, makes a man and a woman. There's a difference. There's only one hydroxyl versus oxygen difference in the hormone. So it's remarkable that the minor difference has a major implication in the in the body. And so usually in a nutshell, what happens that you have ligands, which are the hormones or the growth factors. It could be natural, synthetic, environmental, or it could be in the soil. Long time ago there were some compounds that were in the soil that we were working on were estrogen modulators, and those are all environmental. They were using in pesticides, some of them, and they bind to the estrogen and had a major effect. So all these organic food concept had many of the soil-based problems. These ligands bind to the receptors. Receptors have subtypes, they have isoforms, and they have splice variants. Splices we talked about in nutshell with Nitin's work, and they have isoforms which are I'll talk today. Like for example, ER alpha and beta are the isoforms of steroid receptors. And then you have effectors, your DNA response elements and regulators. They work together in the overall transcription and then on to the action in the in gene expression. So in case of breast cancer, then we have in case of premenopausal women, and then they produce all these different hormones. Now why this is important? Because in the premenopausal women you have the ovaries. Ovaries produce estrogen, and the estrogen works through the estrogen receptor system. Now in postmenopausal women, what happens is that the ovarian function is lost. So you don't have ovarian function, but many times what happens is that they still have the estrogenic activity. So in case of breast cancer, you really have to be careful. That estrogen comes from adrenals, because adrenals produces estrogen synthesis, and that's exactly what they do. They put the aromatase that converts testosterone to estrogen. They will use an inhibitor, which is the first line therapy for postmenopausal women with breast cancer. They stop the synthesis of estrogen. Now estrogen has a function through three different ways. Estrogen can get in and go into the nuclei, and we know all that estrogen response elements work on messenger RNA, generate proteins, and you do the cell cycle regulation and do the tissue growth. Estrogen also has a membrane receptor which would work more like a EGF. So it will bind to its own membrane receptor, estrogen receptor, and do the signaling inside the cell like a growth factor does, and provide its action. And estrogen also has another form. It's a mitochondrial receptor, and that works through the mitochondrial responses. And this is less known phenomenon. It's not been worked out as much in length as to how it affects various things, various regulations in the body or in cancer cells or in normal cells. Okay, there are two kinds of estrogen receptors. We talked about it in Diana's work yesterday, if you remember, the ER beta alpha having differential action. Okay, so you have ER alpha and ER beta. We won't be able to go into this into detail, but all we have to remember is ER beta is shorter than ER alpha. Okay. ER alpha is longer than ER beta by 180 here, 144 here, so it's about 36. You can do the calculation. That many nucleotides shorter. So, but it has a remarkable difference in its activity. ER beta does not bind to ER alpha. They both have differential activities in relation to the growth. ER beta is a suppressor, ER alpha is a promoter. ER alpha, when you have the ER alpha expression, you want to inhibit the ER alpha expression in breast cancer. You want to the expression of ER beta in breast cancer because they have opposite functions. So is a patient ER alpha positive or ER alpha negative? And the cutoff, something that's called ER alpha positive versus something that's called ER alpha negative, is very arbitrary. So what happens is, I'm just trying to tell you like 10 cells or less in the section that are ER positive, then they call ER negative. So if somebody had 8, they are out of luck, which is not good, because you are really trying to sort of take their ability to respond to tamoxifen or anti-SR. After we had proposed in that paper, of course I'm not a physician, can't do anything, but we thought, what about if you give these patients this compound that's going to be an inhibitor of PPA or gamma, that they are used to enhance the level of estrogen receptors. Okay, so this is how the grading occurs. Okay, you have the proportion score, they'll say this is highly positive and so on. Okay, 1 in 100, 1 in 10 cells, 1 in 3 cells, or something like that. Based on that, you can decide whether it's a negative or a weak ER response. I mean, ER is ER positive, intermediate, or strong ER positive. Now there is a direct correlation. So physicians or anybody who is doing the treatment are not wrong, because the more ER you have, the better response you're going to get to anti-estrogen treatment. This is the slide that shows that the ER beta in human breast, if you look at the normal mammary gland or breast, then you have a very high beta expression. But as you have the non-proliferative or get into the atypia or dysplasia or ductal carcinoma in situ, and then you become DCIS, which is a high-grade DCIS, ductal carcinoma in situ, which is definitely going to form do the section and do various things. Now breast cancer is called luminal A. This is the most recent, because until I retired, the luminal A kind, luminal B, there is HER2 enriched, and there is a basal-like. Now luminal is estrogen receptor and progesterone receptor positive, but HER2 negative. HER2 is EGF-like, right? So the growth factor receptor negative. The luminal B is all 3 positive. This is HER2 enriched, which is ER/PR negative and HER2 positive. And these are all TNBC or triple negative breast cancer, right? The 3 different kinds, and these are the biomarkers they use to. First they look at the histopathologically, but then they look. You give the anti-estrogen treatment in premenopausal women. You give the aromatase treatment, aromatase inhibitor, I mean, in the postmenopausal, and they get chemotherapy and radiation anyway. So they start out with this. This is given for 5 years, and then they get chemotherapy and radiation and variety of different chemotherapeutic regimens there. If you have a HER2 positive, you almost always give Herceptin. So here you give anti-estrogens and Herceptin. Now Herceptin was developed by Genentech, and that gives a great response to the patients who have a HER2 positive. So that is, you define the drugs, you have an anti-estrogenic drug that you are trying to develop from the plants, and plants have a lot of them. Many compounds, the anti-estrogenic compounds do come out of the plants. And then, because when we were evaluating those things in anti-estrogenic assay, you get many of them that have anti-estrogenic function. And so if you do a cell culture or in vivo test, and if you find estrogenic activity, you focus on this one. If you do something that is, so you select based on the target you have, based on the target that your drug is going to work on, and you go only after that type of cell, and see which portion of the breast cancer samples that you're going to be working on. Okay, and then what happens is, this is a molecular profiling about 476 samples, and these are all listed here. The red means overexpressing, green means low expressors, and I think that's how it goes. Yeah, above average and below average and not determined type of thing. And the whole thing is the normal is right here, and some genes underexpressed in case of HER2 expressing ones, and some genes are overexpressed in the other kind. And now this is really then becomes a little bit more involved, but also like a fishing expedition sometimes, because you have so many genes, which gene to go after, and that's where as I explained to you yesterday, your effort in the literature, your judicious selection of a gene based on the current literature, based on the data you generate in your lab, you can decide which gene to go after. Okay, so this one just shows that if you have a ER receptor status positive, and we don't know the PR, but the PR is positive, you have 61% of breast cancer will respond to anti-estrogen treatment. Okay, and if you don't have, very few are progesterone receptor negative, but there are some, and the response is less. The progesterone receptor negative tumors are less because progesterone receptor is estrogen receptor target gene. So you almost always have progesterone receptor going up if you have an estrogen receptor high, and many studies have proved that. And then if you have a negative ER, then it's not going to respond to the anti-estrogens. Similarly, here you have the list of HERs, or the HER2 new is right here. It's also called HER2. You can look at this in your, you can read it through. Okay, now I'm going to spend a few slides on aromatase inhibitor. Okay, so for example, androstenedione to testosterone is getting converted by 17 beta HSD, and I've given the long form HSD here. And then you have a testosterone to estradiol is P450, which is a aromatase, which is 19, P450 19. CYP19 is regulating the P450 protein enzyme, that is the aromatase. Okay, that aromatase is required for converting testosterone to estradiol. So the most logical way to do prevention or inhibition of synthesis is to have a aromatase inhibitor, and letrozole is one of the aromatase inhibitor that's in the clinic that all the women get. In our lab, what happened was that we went after this saying that you get letrozole, and letrozole is going to prevent the synthesis, but almost all the women get resistance to letrozole sooner or later. They are going to be resistant. So you need something else that has a different function, which is going to be introduced in the clinic. So here we have a letrozole resistant cell line that Akash had generated in my lab. It took about 9 months to generate a cell line. You have to continuously put letrozole in the medium and try to select out the cells which are letrozole that grows in the presence of letrozole after all the cells are dead. and then you take that and clone them, and you try again letrozole and see how many die. And so I mean it's a fairly long process, and but he did generate. That was also part of his PhD thesis, and then he just kind of looked at the aromatase expressions and things like that. Okay, come on. Okay, and then what happens is that if you do the testosterone, so this is a T47 cell line, which is estrogen receptor positive cell line. These are the vehicle controls, and the trindione, these are all the aromatase inhibitors that are currently used. You have hexamestane, the aminoglutathamide, anastrozole, and letrozole. Letrozole is used as the #1 line of treatment. Hexamestane is used by some people. Aminoglutathamide is taken out of market because of its side effects. It's no longer in the market, and formastane is not really used that much in the clinic. This is the slide I had in there, just to show the confocal microscopy for the aromatase expression in cell lines in the control, in the aromatase transfected T47D and T47 aromatase transfected letrozole resistant cell line. So we had 3 different cell lines to do his work. They move from there to the raloxifene, and raloxifene is used quite efficiently. So now I go into the chemotherapy. Okay. So this session we're going to learn a little bit, very just like a broad view of chemotherapy. Okay, it's a huge big subject, and we answer of that chemotherapy. And the reason is that tumor is a heterogeneous mixture of cells, so it's not going to respond to that. And many times it's non-specific because those are all cytotoxic. Most chemotherapeutic agents are cytotoxic. Most of them are not really targeted to one specific molecule. They work on certain things, like Taxol will work on microtubules, or camptothecin will work on like topoisomerases, but they don't. There is no one, like one effect. It's just over global, like cytotoxic, and so that's why I said it's a marked toxicity. Oh, I'm sorry. Yeah, I was just gonna have 3 slides. This is not the one. I am using the wrong format. That's okay. I opened up a wrong section. But you have the ideal. So what is an ideal treatment? Ideal treatment would be something that's highly efficacious, something that's a tumor specific, that has minimal toxicity. You look at the risk-benefit ratio, which I talked about in previous classes, and the current therapy. I just mentioned that they are not. Oh, what's going on? Oh, there we go. Okay. So one of the things, since it's a botany department, we need to know is many of the known cancer therapeutic agents which are in the clinic and which are highly effective, they've all been derived from the plants. They're all plant derived. For example, vinblastine is from this, and you guys can relate to plants better than me. They have Taxol and camptothecin that came out from the plants, and bryostatin is not a plant, but from the bryozone. It's from the marine thing. But then this was used for some cancers. But 1,000 kilogram of marine bryozone species will obtain about 1.5 kilo of bryostatin. So you need a lot of them. You need to grow a lot of them. If you look at the Taxol and camptothecin, was discovered by our friend Dr. Vani. He had Taxol before camptothecin. He had camptothecin done before Taxol, and Taxol was already there, but he did not take it out because he thought that, well, he wanted to put camptothecin, and then they did the Taxol, and the whole world went after Taxol. That's one of the best chemotherapeutic agent right now in the market which works on many types of cancers. It can work on the receptor activities. We covered many of these in our last 3-4 days. We talked about the inhibitors of tyrosine kinases. We talked about that. Okay. You have various classes of drugs available in the literature. You have alkylating agent, you have platinum compounds, you have anthracyclines, you have antimicrotubular agents, antimetabolites, and topoisomerase II inhibitors. So, for example, the ones which are like Taxol is an antimicrotubule agent, and there are cisplatinum is a platinum compound, and some of these compounds, for example, there is some sort of a selectivity. So when they look at the combination chemotherapeutic regimens, what the doctors do is they have a combination protocol already established. So they will have the adriamycin for breast cancer with a couple of other drugs, or they will have a Taxol as a second round of therapy. They will give cisplatinum to the lung cancer patients, things like that. So they have dose of the active agent in order to reduce the toxicity of that agent 1 in order to be able to introduce agent 2. So the way it is done is, and somebody had asked me a question, I forgot, is how do you do that in the animals? And the way you do is you identify for one drug, for both drugs individually, the most ideal dose, that reaches to a toxic level. And in this one, toxicity is allowed. So you can allow up to 10% of body weight loss. So you can give the compound and give in increasing concentration, 5 animals per group, don't need too many, and weigh the animals daily. And at the end you can look at the liver profile or whatever you are doing, and then see what concentration the animals start losing weight. The other way of doing it in the experimental condition is you use 1:1 ratio, you use 50:50 ratio, and see how they are going to pan out, and then you can select, you can titrate your doses based on that. Okay, you have you want to have a cure, but it's just sort of difficult. But then you also have different types of treatments now people are doing. One of the most popular one is called neoadjuvant. So what used to happen is, and in case of breast cancer, is as soon as a person is diagnosed with breast cancer, the psychological trauma is so much they want to get rid of it right away because it's a cancer. So the surgery was first action. It still is the first thing that they do. They remove the tumor, and then they start the radiation therapy and the chemotherapy. And these are not fibroadenomas. Remember, fibroadenoma is class number 1, where you have encapsulated tumors so that they can remove the tumor and it comes out as the entire piece. Here it's not going to happen. Here they have to remove the tumor very carefully. There will be odd shapes and they won't have boundary, and so they remove it. The cells get dispersed, and some of them may get introduced into the blood vessels, which is very likely. And so what people started doing was doing a neoadjuvant therapy. So they give, before they do surgery, they give 1 or 2 cycles of chemotherapy. So the cells are going to stop dividing. The cells won't grow. So they are becoming dormant. So now the cells, when you take it out, the cells are in a dormant condition. You take them out and then you give chemotherapy. So you kill the cells that are left. That chemotherapy is going to kill most of the cells. They will see if chemotherapy works or not, if the tumor shrinks or not, and then they do the surgery. And their outcomes are much better. So many hospitals, it depends really on the cancer, and then they will give a chemotherapy and then go into the surgery. So there is not too much to remove also, right? Okay. The clinical trials are divided into 3 phases: phase 1, phase 2, and phase 3. phase 1, so when your drug is approved, it's always approved for the phase 1. You have to kind of give them the data back and forth. The phase 1 clinical trial is just to see the tolerability. So what they do is they take the patients who are advanced with different types of tumors, breast cancer patients, if you can find, that's great. And then they do the optimal dose, what dose is tolerated by a patient. So that's phase 1. So all new drugs have to go through that. Every drug has gone through, but people like us who have new drugs that we are proposing to go forward, like D5, when we get the approval from the FDA, we can set up a phase 1 clinical trial. They will allow us to do it. Phase 2 clinical trial assesses the tumor response. So the treatment is actually doing, but what they do is they already continue the treatment which is already going, and then they add on top of that something like this. And phase 3 is a huge big randomized trial and requires millions of dollars, and the academicians cannot do it. You have to have it done by the industry that has a lot of money. So, and we had a discussion in the hallway sometime that people are that the pharmaceutical companies are introducing drugs because they want to and fail. But all those trials that failed was expensive. So somebody foot in the bill. So what they do is they try to make it up for the money they lost, essentially, which is not good, because a drug synthesis or production may not cost as much as it costs for the treatment. So it's started androgen therapy. The ones that are androgen receptor negative, they get this other therapy, non-hormonal therapy. Then we talked about the aromatase inhibitor therapy and so on. This is the Herceptin. This is a humanized monoclonal antibody to HER2 receptors, and they are given. They have some side effects. There is cardiac toxicity, but you have no. That is the. So here you are talking about the risk-benefit ratio we were talking about. There are all the risk for cardiac toxicity. If a patient is a HER2 new positive, they definitely will give him, Herceptin. That's what's given right now. Okay. Then we have other targeted therapies, like we have a VEGF the targeting, we have VEGF receptor target, we have HER2 target, and many of these clinical trials are ongoing with the drugs like this. These drugs are the antibodies. When it has a MAB, ZOO means humanized, one gene panel. So over all this experience, physicians, not the physicians, but the labs, in the lab scientists have come up with a 21 panel gene, and it's called Oncotype. And they run the whole genomic gamut, and they find out whether the person has a cancer at a very early diagnosis. And it's been very effectively run. And I was talking to Dr. Rahul the other day, and he said that this assay, I asked him that do you do the Oncotype, and he says that no, that somebody had it, he was telling somebody. So they had it, and he says, well, it costs 1.5 lakhs for one sample for the identification. And I don't know why. Actually, that's not a bad idea. I should open up a lab for Oncotype and introduce it in India, and I can probably do it in $1,000, right? Which is 50,000, 60,000. I don't know, because once you have a chip is available commercially, all you do is do RT-PCR on a chip. It's not. It's not a rocket science. It's very easy. Okay. So this Oncotype assay, the result comes out as a R score. There is a formula, a software, and it generates a score. So if you have a less than 10, you have a very good response rate, and if you have R score of 25 or greater, then chemotherapy and hormone, depending on the ER positive tumors, or ER positive and node negative. So your nodes have been shown to be negative from each patient, whatever patient comes out, and cancer cells, they will grow many generations, and they'll put a panel of chemotherapeutic agents in the culture and see which one gives the best response and treat the patient with that therapy, because it's a personalized medicine. Then this happened about 15 years ago. That's where age helps, because you remember what happened. So 15-20 years back, this person was working at NIH. He came up with this idea. He opened a company. He left NIH and cycles DNA, high proliferation. So as we have to wrap up a little bit sooner than previously thought, so I was going to give you the entire cancer prevention and therapy overview. But we have talked about vitamin D throughout the presentations and went through MMOCs and different cells and stuff like that. So I'll switch gear and I won't talk about vitamin D at all, and I'll talk about our work on Dagulin, and that's on the therapy part. And Raksha has done quite a bit of that work, so I'll show you her data from our lab. Thank you. So now you can have tea. No, synthetic. One of the problems, one of the reasons it's so expensive, that it's the organic chemist always calls number of steps. How many steps to synthesis. So Taxol is a huge number of steps. I don't know how many. I used to remember, but I don't remember now. But what happened is that the company that opened up in USA, they specialized in the synthesis of Taxol, Taxotere they call it. It's just a new generation compound, and that has material as your control. And then you run your assays with the synthetic compound. Then you find that synthetic compound is as good as your plant derived. Then every time a new batch.
Taxol is
Yes.
Taxol is normally produced by an endophyte rather than the plant itself. So I was.
My impression was that it comes from the
secondary metabolites.
Taxol brandfolia.
It is from plant.
There was a controversy that it is not produced by Taxus brandfolia, but there is an endophytic fungus in the plant which produces it.
Ma'am, actually there is a horizontal gene transfer, I think, from plant to the microorganism or a fungus that is responsible for the production of that.
getting screened in the chemotherapeutic assays. So when they do these assays, they identify something that is very active, and then they go after that. So Taxol, when it first came out of Vani's lab, it had gone through like 5 or 6 different screen assays in Kinghorn's lab, and it was done. Now when this was identified, I remember that the National Cancer Institute said that they have done thousands of compounds, and they were very happy if even if 1 or 2 shows the positive side. Now there is a repurposing of the drug program, 300 some odd compounds over a 3-year period in nude mice. And in my lab, came to me and says, well, we found a drug. So this is it. We're perfect. So it's okay. Let's see that. So it had a number. So we ran the number entry into the NCI thing.
People identified that fungus
Produces.
Right.
Maybe in symbiotic association.
These are just the studies I'm sure somebody must have done. But, I'm not in the plant biology. Like making wine, red wine, which will have very high resveratrol.
We talked about the personalized medicine in the patient based upon the genetic.
Wants in terms of growth, cell proliferation, and the target was not really a focus. since it's becoming personalized medicine, the target, everything becomes molecular. The target is important, and they do try to find out the target.
So, why some people are more prone to certain type of cancers than others? Like for example, when we talk about prostate cancer, we always say that males of the Caucasian race are more prone to prostate cancer than of the Negroid race.
That's not true.
What makes them more prone?
That's not true.
That's not true.
No. No.
So, like, there's no truth about the fact that some.
No, there is a truth about race. You are right for many things, but this is not true. What you are saying, we have seen reports, lots of reports that the blacks have, have.
Demo resistant.
Yeah. No, they do have very high incidence of prostate cancer also. But the whites also have it. But the prostate cancer is more of age related. So, the androgen independent cancer if you get, this is like you give flutamide and things like that. But to the extent now that if you have a hyperproliferative prostate, right, the physicians don't even remove them. They just kill that with the radiation.
India, we have food extract that's given to the patients. Now, most of the time we feel that the synergistic effects of a lot of molecules within that gives a better output. I mean, the pain is less, the tumor shrinks faster.
I'm a believer in that too. Yes.
So I have also seen that a lot. So I feel that Ayurveda and Siddha have some say when it comes to cancer,
Right. What happens, I tell you what's the reason. The reason is that the science has become more as perfection. Right? So like as I was just telling you, that when you do even you test a compound, with proper control to see how well it compares with the previous data. Now in Ayurveda, you're going to have the herbs or plants or leaves or whatever you grind up and mixtures. The temperature is going to make a difference, season is going to make a difference, drought is going to make a difference. So your pool of samples that you are going to do is absolutely not going to be homogeneous. It's going to have a lot of variations. And scientists or NCI are very reluctant in doing that. That's why from green tea, GCG was isolated, because they had found that green tea is really good. But then there was a thing that people who are very rich, they will use every cup will have a new brewing of fresh green tea, and like me, who may not be making too much money, you just try to use the same thing for 50. Or in the college, I was in Tokyo University. I was a fellow for 6 weeks, learning about it. And you had I said, where do you change this green tea pot? And I said, no, you just put the fresh water and there it goes. So that effect of that tea is not going to be the same as the effect that the king is going to drink. So those are the reasons this natural or adaptable something to make it work, and that may be something in the whole food.
Sir, is it safe to assume that all the compounds in a food will be safe?
Well, you are eating the food.
I was working in glucosinolates, and there are two specific isothiocyanates which are progoitrin and goitrin, usually called goiter. But other isothiocyanates in the mixture are very nice. So I think whole food mixture is good, but at some point, because it will cause some harm or other things.
True. So for butyl isothiocyanate, you have to take in large amount to get a goiter. That means you are taking
samples coming from all over India will be very less, because no one can afford this kind of.
So why don't they make the chip here in India?
So we are working with iron tolerant. So iron tolerant, we don't use the micro iron chip or anything. We are using the.
Individual RT-PCR?
Yeah, that is only for authentication later. We don't do RT-PCR. The instrument run is somewhere around 40 to 50 pounds. The instrument run, single run will be possible.
that I don't know. No, I know that's not.
Yeah, that's the bad part.
Experimental design problem later on in your labs. Just drop me a line and
You can always communicate.
Yes.
Regarding
I am known to criticize everything, so
planning any kind of. We have made the certificates, and since Dean Academic appeared in Kochi, so we could not get the signature. And we have put our signature, but Dean Academic signatures they are also very much required. And we will distribute you the certificates in front of Vice Chancellor. You take the certificates, and afterwards you submit it to these two students, my two students, Pradeep and Sadeep. Okay, and most. आपका नाम लिख देना राइट फीडबैक फॉर्म में भर दिए आपने या सबमिटेड मैडम ने चिंता नहीं दे फीडबैक बहुत बीते बच्चे No talking, no talking. Please. Learner person.
अभी नहीं करना है আমি তোমাকে চাই, কাপ চাই, চাই, আমি তোমাকে চাই, কাপ 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 চাই, কাপ চাই, আমি তোমাকে 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 চাই, চাই, আমি তোমাকে চাই, কাপ চাই, আমি তোমাকে চাই, আমি তোমাকে চাই, কাপ 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 চাই, আমি তোমাকে চাই, আমি তোমাকে চাই, কাপ 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 চাই, কাপ চাই, আমি তোমাকে 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 চাই। কাপ চাই আমি তোমাকে চাই 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 কাপ আমি তোমাকে চাই কাপ চাই আমি তোমাকে চাই কাপ আমি তোমাকে চাই কাপ চাই 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 আমি তোমাকে চাই কাপ কাপ চাই আমি তোমাকে চাই কাপ কাপ চাই আমি তোমাকে চাই কাপ চাই আমি তোমাকে চাই কাপ চাই তোমাকে চাই কাপ চাই আমি তোমাকে চাই কাপ চাই আমি তোমাকে কাপ চাই আমি তোমাকে চাই 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 কাপ চাই